This is the second video of Chapter 3. In this video, we're going to be focusing on the cell surface. We're going to talk about the structure of the plasma membrane, the components that make it up. We're going to talk about a really important system called the second messenger system. It's super important in human physiology and also drug development and pharmaceuticals. We're going to define the glycocalyx and talk about what it does, and then we're going to talk about a few ways that cells can move. So we've talked briefly already about the plasma membrane. This is the border around the cell that separates the inside of the cell from the outside of the cell. When we look at a micrograph, we can see that there are two layers. So this is one layer of the phospholipids. We have the hydrophilic heads pointed towards the outside of the cell, and then an interior made up entirely of the hydrophobic tails. They're made of fatty acids. And then on the inside, we have another layer of hydrophilic heads. So the head portions are oriented towards the aqueous interior and the aqueous exterior, and that layer in between them is hydrophobic. Water doesn't like to go there. If we have two cells that are close to each other, we can see they each have their own lipid bilayer. So the green is the lipid bilayer of one, and the pink is the lipid bilayer of the other. We have the intercellular space inside and the extracellular space on the outside. And here we have the intercellular space of cell 1, the intercellular space of cell 2, and the extracellular space between the two. Here's a cartoon of what the lipid membrane looks like, that plasma membrane made of phospholipids with proteins embedded and other lipids incorporated. If we were to count the number of molecules and identify them as either being lipids or something else, 98% of the molecules would be lipids. Only 2% of the individual molecules would be proteins or carbohydrates. Of all those lipids, 75% would be phospholipids, and they're arranged in a bilayer like we were just talking about, with the hydrophilic heads pointed towards the extracellular matrix and the intracellular matrix. It's important to note that these phospholipids kind of move like oil on the surface of water. They're drifting back and forth. They can spin around in place. One phospholipid can trade places with another. This is a very fluid kind of flowing thing. 20% of the lipids would be cholesterol. And cholesterol's job, remember, cholesterol has that more ring structure. I told you guys I like to think of it as a chain link fence. It's a lot more stiff than the phospholipids, so it can maintain the integrity and the shape of the cell membrane. And then the remaining bit of the lipids are glycolipids. Okay, so these are phospholipids that have carbohydrates attached on the extracellular surface, and we'll talk about these more when we talk about the glycocalyx later on. So we said that 98% of the molecules in the cell membrane are lipids. So that means 2% are proteins. However, those proteins are so much bigger than any individual lipid that the proteins make up 50% of the weight of the membrane. So there's fewer of them, but they are much bigger. So we have two general types. We have integral proteins, which are also called transmembrane proteins. And I think transmembrane is easier to relate to what they do. Okay, so a transmembrane protein is a protein that goes all the way across the membrane. You've got some of the protein sticking out into the extracellular matrix and some of the protein sticking out into the intracellular matrix. So it goes all the way across that phospholipid membrane. Remember we talked about proteins fold up into the complex three-dimensional shapes, and they want their hydrophobic regions away from water and hydrophilic regions close to water. And that's how transmembrane proteins fold. Transmembrane proteins are going to have hydrophilic regions that are sticking out on either side of the plasma membrane, either into the extracellular fluid or the intracellular fluid. But the portion of that transmembrane protein that is actually stuck into the phospholipid bilayer is hydrophobic, because those fatty acid tails are hydrophobic. So, like the phospholipids, the proteins can kind of drift around just a little bit. They're generally anchored by cytoskeleton inside the cell, so they drift less and have less freedom of motion than the phospholipid bilayer. In contrast to the integral transmembrane proteins, we have something called peripheral proteins. Peripheral proteins don't go all the way through. They're only on the outside of the cell surface. So we have a depiction here of the transmembrane proteins, and they have their hydrophilic region in the aqueous extracellular matrix. They have their hydrophobic region stuck in here with the hydrophobic fatty acid tails. And then they have another hydrophilic region protruding into the intracellular area. There are a lot of different functions of these membrane proteins. They do a lot of different jobs for us. Okay, so they can be receptors. They can take in molecules, and those molecules may, once they're properly positioned into the receptor, change conditions or cause the receptor protein to take some action on the other side, causing the cell to see an internal change. These can also be enzymes. We've talked about enzymes quite a bit. So the enzyme can come into the active site, and while it's in the active site, some sort of chemical reaction takes place, and the products then move along away from the protein after the reaction is complete. Some membrane proteins are a channel. It's like a tunnel. Whatever the thing is, in this case we're showing some ions, the ions can just pass right on through. They don't have to fight their way through the plasma membrane. It's like a tunnel. The ions can just flow on through, in or out, according to their concentration gradient. We also have gated channels. Gated channels can open and close with various cellular conditions and allow the ions or whatever other chemical we're talking about to come through only at certain times. We have cell identity proteins, and these are things that are going to allow the cell to recognize itself, its own cells, versus invader cells. So think of your blood type. You could have a type A marker here, and when anything other than type A blood is encountered, it could cause an adverse reaction as the body recognizes the transplanted blood cells are foreign. They are not self. And finally, we can have membrane proteins that are involved in cell adhesion, where one cell attaches to the other, either long-term or short-term. There's a very specific class called second messengers, and these are super important. The reason they're so important is because 60% of the medications that we take work by this mechanism. So these are really, really important for us. Let's talk through it, and I'm going to go into a little bit of detail here. So if I say words that you aren't familiar with, Google can be your best friend on this. If you miss a word or if you haven't heard a word that is part of this description. Okay, so here we have an integral or transmembrane protein, all right? It goes into the extracellular matrix and also into the interior of the cell. And we have a chemical messenger come in, okay? In this case, our example chemical messenger is epinephrine. Okay, epinephrine is the chemical name for what we might more commonly call adrenaline. Okay, so we have an epinephrine coming in, binding to the receptor, and that's going to cause a change. And in this case, the change it causes is the release of this G subunit that's attached to that integral protein. So now this G subunit is free. It's traveling in the intracellular matrix inside the cell, and it comes into contact with another transmembrane protein. This one is called adenylate cyclase. Okay, so adenylate cyclase is impacted by the G protein, and this causes a reaction. Okay, so we have ATP that is going to be converted into something called cyclic AMP and 2 of these inorganic phosphates. Okay, so we've talked about ATP. ATP is adenosine, a sugar, and then 3 phosphates all put together. And those 3 phosphates all have their negative charge, and so they don't like to stick together, and energy is released when the phosphates come off. So the T here stands for triphosphate, 3 phosphates. Here we have cyclic AMP. Okay, so cyclic means in a circle, in a cycle. So the shape changes and it's a little bit different shape. Here M means monophosphate. Mono means one phosphate. So we went from 3 phosphates to 1 phosphate with 2 free phosphates. So all our phosphates are still accounted for. Okay? Getting a little deep into biochemistry, I know, but this one is super important for you guys to understand. Okay, so now the cyclic AMP is going to be able to do lots of different jobs in the cells. And depending on the specifics, this is kind of a generic example, the specifics here, we can target certain reactions in the cell that can fix different disorders that a person might have. So we've talked before about an enzyme called a kinase. Okay, I mentioned it briefly in a previous video. Kinase is a general term for a whole group of enzymes. There are lots of different specific kinases, but as a group, kinases add an inorganic phosphate to something. Okay, so the cyclic AMP activates the kinase, and then the kinase does its job of adding inorganic phosphate to some enzyme, and that allows a process to start. The glycocalyx is a coating that you find on the outside of the plasma membrane. So it's on the outside of that cell membrane there. It's made up of carbohydrates and also some glycoproteins, which means carbohydrates and proteins put together, and also some glycolipids. So here we're looking at carbohydrates and lipids put together. Every single person has a unique glycocalyx. Every single person has a unique combination of carbohydrates that allows the cells to identify themselves as self and not foreign. The exception is identical twins who have entirely identical DNA. Okay, so outside of identical twins, everybody has a unique glycocalyx, a unique carbohydrate signature on the outside of all of your plasma membranes. This glycocalyx is involved in your immune system. It recognizes anything that is not self. It has a role in preventing cancers. Obviously, when we do organ or tissue transplants, we need to consider the glycocalyx in order to make sure we have compatible tissues being transplanted. This also has to do with fertilization, because when a sperm and an egg join together, that fetus's DNA, the embryo and then fetus's DNA, is different from the mother's DNA. And so we need to involve the glycocalyx in not identifying the embryo as a foreign invader to be destroyed. Now we're going to talk about some of the projections that we can find on the exterior of the cell. So the microvilli is one. Microvilli are used in absorbing nutrients. So the entire interior of your intestines is microvilli, and they allow the nutrients to be absorbed from the food you eat as it's digested. Cilia are another type. So cilia have a few different varieties. There are some that are stationary. They don't move. They just stay in place. And they're like antennae. They are collecting information about the external environment around the cell so that the cell can respond properly. There are cilia that are able to move. A really good example of this is in the fallopian tubes. It's cilia that help the egg to move from the ovaries to the uterus. Next, we have flagella. The only true flagella that humans have is the tail of sperm cells. Other species have more flagella, especially single-celled organisms. If they're able to move, oftentimes it's by flagella. We can also have flagella, we see them here, these longer tail-like structures, along with cilia. So this picture shows both. Flagella are used for movement, and I think you guys have seen like videos of sperm cells swimming at some point in your life. They kind of move like a fishtail. And finally, we have pseudopods. Pseudopod means pseudo is false, and pod, think of podiatrist, pod, foot. So false foot. These are, as a cell, not something that has feet, but these things kind of extend out, kind of like feet look like. They can be used for two different things. The first one is moving. They can kind of reach forward and pull the cell along. And they can also reach around different foreign invaders and engulf them. And this is how some of your white blood cells work. So that's a quick overview of the plasma membrane and some of the proteins that extend through the plasma membrane, with a closer look at secondary messenger systems. And finally, some of the different projections we can have from the cell surface.